and there's electronics, and all of those things go together so that the numbers that we get out of the instrument are something that we understand and can trust. So let's talk about gas exchange. How do we measure gas exchange? Well, we use an erga, which you will also see called non-dispersive infrared, or NDIR. For those of you who use the LI 6400, you might have occasionally seen ergas not ready. And what that meant was your instrument was telling you that something about the infrared gas analyzer was not working. And it might have been something related to your cable or your optical path or something, but whatever it was, the basic measurement was not working. And the principle of a basic optical differential infrared gas analyzer is that we have a source, which is an infrared light bulb, essentially. And then we have an optical path. We have a detector. And what we do is we measure, we know how much light went in. We know what that number was because we know what the light bulb qualities are. And then we also just measure what comes out. And the difference between what we put in and what we put out is tells us that that light was absorbed by a gas. And most of our infrared gas analyzers have an absorbing region and a non-absorbing region. So what that does is tells us what the background absorbance is, the dirt, the dust, everything, versus what is actually CO2. So or H2O. All of our instruments that are built like a CO2 absorbing region and a non-H2O absorbing region and a non-absorbing region. And that chopper motor is constantly spinning to allow us to understand what in that air is CO2 and water and what in that air is not. Based on that absorbance, we can start to use the electronics to determine concentrations. Pardon, T is very important when talking. We put the infrared gas analyzer, the erga, into a gas exchange system. And gas exchange system, so there's the chamber, we put the leaf in or the other material in the chamber. And from there, we calculate the amount of CO2 that must be going into the leaf because it is a closed system. It has to go somewhere. And that was the principle of the LI 6000 and the LI 6200, if any of you have ever used that. The challenge with a closed transient system is that we can end up with some physiological confounding factors. Is our plant responding to the change in CO2 because of a system? Is our plant responding to the fact that the CO2 in the chamber is dropping? And so as a result, there might be changes in stomatal behavior, there might be changes in Rubisco behavior. All of these things might be happening. Some systems are closed, but you add a little, so periodically we just give it a little bit more CO2 to try to make sure that a little bit of CO2 is taken up by the leaf, but then we give a little bit back. After open flow system to the chamber always has In this situation, we're not as we kept chamber volume. In an open system, we're more concerned about flow rate, how quickly that air being exposed to the leaf, how quickly could the CO2. In an open system, we have a mass balance. The idea is that we have the same amount of air going in versus coming out, although we might have different concentrations of CO2 and water in the going in versus going out. We hope so anyway, since the basis of our measurement is that there's a difference between the two. But versus flow rate going out, we measure that. We measure water going in and water going CO2 essentially, and then the difference between the two tells us either transpiration or looking at change in CO2 something's always happening, but we don't know because that information hasn't reached us yet. So in the LI6400 and LI6400 XT, what we did was we moved the analyzers to the sensor head. And we said, okay, let's just make sure that if it's happening at the leaf, we know about it immediately. So we moved the analyzers to the leaf, and that meant that any response time was much faster, and we were able to understand what was happening at the leaf and respond to that quickly. The flow meter was still in the console. And we had two tubes that we split the air to go to sample and reference. In the LI6800, we changed it up a little bit even more. We moved the flow meter to the sensor head. Excuse me, we have one line of tubing going from the console to the sensor head, and we moved the location of the analyzers. It doesn't really affect the response time, but it did help us out in a couple of different ways that we'll see a little bit later. So it's pretty much the same as the LI6400 XT, but with some improvements in terms of the response time, diffusion characteristics, and instrument behavior. So here is our view of the console. So if you could take the console apart, which you should not do, then you would see that we have air going in, we have an air filter, and we have a soda lime column, which takes out CO2. Here's our pump. We have a humidifier column that is either Nafion or Stuttgarter granules. The Nafion is the new one, and if you don't have the new Nafion tube, we will talk about that later and why you might want to think about getting it. And then we have the Dryrite or silica gel, which removes water. So soda lime removes CO2, Nafion adds water, and silica gel removes water. Why do we have a tube that adds water and a tube that removes water? Well, that allows us to make sure that if we ask for a certain water concentration, we get it. We say to our leaf, I would like you to have, we say to our instrument, I would like the leaf to have this much relative humidity. And what we can do is we can use these two columns together, add a little bit here, take a little bit there. These little bow tie looking things are valves, and they allow us to either turn it on, turn it off, and then also open and close that valve. We also have the same for our CO2 cartridge. This is an 8-gram CO2 cartridge, and that also is something that we can open or close the valve to make sure that we T-shaped thing is the auxiliary input port. So if you don't want to use the pump, but you do want to supply a gas, maybe you're working in a lab and you're working with a certain concentration of CO2, you can do this. You can do an auxiliary input of air. When we go to the sensor head, we have this view. We have the leaf over here. We have a mixing fan, which we'll talk about shortly. And then we have our two analyzers, our two ergas. We also have a series of bow ties valves here, and this allows us to send all the air to one of the analyzers or the other, or usually we mix them. So we want to have air going to both analyzers so we can measure them, but we might change that proportion of how much goes to one versus the other. We have flow meters that are on the external. We have a main flow meter and we have flow not actually shown here. And we have two sample and reference removal, sample removal spots, so that if we wanted to go to a GCMS or an isotope analyzer, another instrument, we can pull air from either the sample or the reference at the back of the sensor head. And we'll show you the actual physical parts of this a little bit later, but this is the general diagram of how it works. One of the things that that series of valves here and the ability to move air through and around the different analyzers allows us to do is what's called matching. So we have two separate independent analyzers, and they might occasionally get a little bit off from each other. You know, one of them thinks it's 399 and one of them thinks it's 397. Well, that can cause an offset in apparent photosynthesis. We don't want that. So what we do when we match our analyzers is we say, okay, I'm going to flow the same air through both analyzers and make sure that both analyzers are reading the same information. Matching is one of the most important things you'll do with the instruments, because without that, you might discover that your analyzers have been wandering off from each other, and it just adds too much complication to life. So when we match the analyzers in the LI6800, what we do is we run the same air through the reference and the sample. In the LI6400, we needed the leaf to be stable so that we could compare the measurements between reference and sample. In the LI6800, we just ignored the leaf for a little while, and we use our valves to send the air through both analyzers, but not through the leaf chamber. We just ignore that. This allows us to match before the leaf is stable, and it also allows us to do a few other things related to matching. There are two different matching options in BlueStem 1.4 and after. We're now at 1.5. There's point matching, which is what we might call the old standby, which is that we match at a certain CO2 concentration, and if we change CO2 concentrations, we might need to match again. Or there's range matching, which is where we match the CO2 or the H2O at different ranges, and then we have that information in the instrument's memory and can use it. So yes, point matching, analyzers are matched at the current CO2 and H2O concentrations. This is the way we've always done it. Range matching, we use data over the range of CO2 or H2O or both to determine future match offsets. So if I change my CO2 concentration, I don't need to match again because my instrument can just use that range data that it had. It must be done when the analyzers are completely warmed up. Otherwise, the range is not going to be as large as it could be. So you want to have your analyzers warmed up for at least an hour before you do this. You can do